Hi everyone, my name is Moon and I'm a postdoc at the Yale Science Building. In this video, we will talk about a method called in situ hybridization. It is also abbreviated as ISH. So this method helps us to detect the presence or absence of RNA in certain cells or tissues. The words to be remembered or emphasized are RNA, and we are detecting it in cells or we are detecting it in tissues. So how does this method work? Let's decompose the word in situ hybridization into two parts. The first part is in situ, which means on site. So this method not only lets us identify the presence or absence, but also helps us pinpoint the site of expression of RNA molecules. The second component, hybridization, simply means mixture of two species, just like the mythical character of chimera. The first component of our chimera in context of ISH is a string of DNA or RNA nucleotides of interest. The second component, also called as probe, is a string of nucleotide of RNA complementary to the nucleotide of interest. We let these two components fuse or anneal. The resultant is a double-stranded hybrid molecule. Now, why does this annealing happen? This happens because of complementarity of bases. Now let's zoom into this small region of our hybrid molecule. So say the sequence of this nucleic acid is UAGC. So our designed probe will have a sequence of ATCG. This is complementary. When they find each other, they will hybridize and they will make our chimera. So how do we detect this chimera? There are a number of ways. Here we will label our probes with fluorescent molecules. This labeling of the probe will help us detect with fluorescence the hybrid molecule. Now let's put all of this into context of an application. In this class we will understand what gives a cell its identity. So what makes a pancreatic cell or a liver cell different from all other kinds of cells? So remember, liver cells make albumin while pancreatic cells secrete insulin. Recall that the gene of both albumin and insulin are present in both liver cells as well as the pancreatic cells. However, as the cell adapts its identity, it transcribes into specific RNA. The genes transcribe to RNA while the RNA make proteins. So the liver cell will have abundant of albumin RNA, which will make albumin, and the pancreatic cells will have abundant of insulin RNA, which will make insulin. Now let's think about a hypothetical situation. Say your friend confused between two slides. The slides either had liver cells or they had pancreatic cells. You need to come up with an idea to solve this problem. You jump in and suggest to do ISH. You design a fluorescent probe, which is an RNA molecule, against albumin or insulin. You add it to the cell and let them anneal. You wash the slides to remove any annealed RNA. You observe the slides on fluorescent microscope. The liver cells have signal for albumin and the pancreatic cells have signal for insulin. Now you tell your friend to label the samples next time. Thank you for listening to the ISH review.